I think we have to move on to our next speaker. So our last speaker in this session is Abdellaoui, and he's from the Amsterdam University Medical Center.
Okay, thank you. Thank you, Ian. Thank you, NIH, for inviting me to be part of this amazing crowd of people. I'm going to tell you something about genome-wide association studies. It didn't take long before we realized that sample sizes are key in increasing genetic signals. So appropriately, the sample sizes have more than tripled over the past 5 years. But we are all gathered here to try to understand the content of those signals. So what we did here, we downloaded all of the GWAS summary statistics from GWAS catalogs, or thousands of summary statistics, and did some QC filtering. The strongest filter was on sample size, so it had to have at least 50,000 participants in order to get some decent estimates for of genetic correlations between traits, because we made a very big genetic correlation matrix with all pairwise genetic correlations between all traits, and then did a principal component analysis on those on that matrix to see what the largest patterns of variation are in all of these GWASs that we are conducting. And these explain the most variation. These five, I think, around 35% of the variation. And this first principal component explained 10% of the variance. We called it cognition and socioeconomic status, but the strongest loadings are for these GWASs, actually, educational attainment and income. Mostly educational attainment. Income, there aren't that many GWASs of that, but that shows a genetic correlation of larger than 0.9 with educational attainment. And this is a very interesting signal that's so ubiquitous across all these GWASs. And if you look at processes that, things that influence the genetic makeup of a population, so things like migration, assortative mating, fecundity, how many children people have. These signals are most strongly associated with those kinds of outcomes. But what do these signals contain exactly? I mean, we know there are no genetic variants that encode for your diploma or the money on your bank account. These are these genetic effects travel through very low-level biological processes that we talked about all day yesterday, and those influence traits, and those traits influence each other and influence what environment you are exposed to, and that environment influences you and those traits and perhaps those biological processes, and all of that gets captured in the GWAS signal. So the stack of turtles that Xander showed us yesterday, that's been mentioned a couple of times today. Well, that figure right there, that is our stack of turtles. And this paper helped expose the turtle on top. So that was not what we set out to do. We tried to look at the geographic distribution of polygenic scores. So we collected GWASs on a wide range of traits: physical, mental health, behavior, personality. Any GWAS that we could get our hands on that was big and did not include the UK Biobank. We used to build polygenic scores for the 450,000 individuals that fall in the European ancestry cluster in UK Biobank. And then we looked at the geographic clustering. So here on the x-axis you see, let's see, can I get the arrow? On the x-axis you see the Moran's eye. That's a measure for geographic clustering, this spatial autocorrelation. And before controlling for ancestry, a lot of the polygenic scores showed substantial geographic clustering. A lot of it looked like those beautiful geographic distributions from the PCs. Those reflect ancestry differences within the country. Often they are in line with geographic barriers or cultural barriers. Here, often you see the difference between Wales, England, and Scotland. We're not sure why many of these polygenic scores showed these distributions. It could be a lot of the things that Bogdan talked about, or maybe there are actual differences between those ancestries, or the GWASs did not control well enough for population stratification. But that's not what we were interested in. So we controlled for the first 100 principal components, and then we saw all of the geographic clustering drop, except for one polygenic score, and that was educational attainment. And when you plot that on a map, you see the same map as the socioeconomic differences in the country. So you see the Townsend index next to the polygenic score distribution, and those black lines, those are coal mining regions. The coal mining industry collapsed between the 20s and the 80s in the past century. A lot of joblessness in those areas, and a lot of environmental stressors. It's very different to live in those regions than in the rest of Great Britain. And I see half of this figure dropped, not sure why, but what it shows is that people that were born inside the coal mining regions have a higher polygenic score on average than the rest of the country. And this figure actually shows the same, but longitudinally. So we split them up into different birth cohorts. And you see this red line, these are from the people born in the coal mining regions. That drops faster than the blue lines. And this green line here, that comes from this red line. So there's they show consistently a higher polygenic score than the rest of the country. These are the people that migrated out of the coal mining region. So there's sort of a brain drain going on that is detectable on the genetic level that is increasing these regional differences. And this shows similar, this is another way to show the effect of migration. So we compared birthplace with current address. And you see that the variance explained by region increases when you look at current address compared to birthplace. And for the principal components, so those older ancestry patterns, those decrease. So you have those old geographic patterns of ancestry. Those get broken up by these more recent migrations that are driven by socio-economic forces. And just 2 weeks ago, this preprint came out that did exactly the same analyses in the Estonian biobank, about 180,000 genotyped people, about 20% of their population. And they see exactly the same thing happening. So the geographic clustering increases because of the migration for the polygenic score of educational attainment, while for the PCs, for all of the PCs, the geographic clustering decreases, so that those older ancestry patterns get broken up. And if you plot the polygenic score for educational attainment in Estonia, you see that this is largely driven by these two university towns, Tartu and Tallinn. And they also looked at a lot of polygenic scores. We looked at 33, and they looked at more than 160, I think, and they saw the same outlier, the one for educational attainment. And here on the x-axis, you see the correlation between the other trait and educational attainment. And the higher the genetic correlation for the other trait with educational attainment is, the stronger the geographic clustering becomes. We saw the same thing in UK Biobank. So here on the x-axis, you have the measure for geographic clustering. And on the y-axis, the absolute genetic correlation with educational attainment. With intelligence, that's about half of that signal here on top. And so this GWAS on educational attainment, it captures this collection of traits that influence how well you do in school or how well you do in society. And all of these traits get clustered not randomly. As I said earlier, it's very different to live in a poorer region of the country than in the richer region, especially in the UK, which is the most unequal country in Northern Europe. And here you see the geographic distribution of clearly an environmental variable, the number of fast food restaurants in the region, and that shows the same distribution as that polygenic score. So you get genetic effects and environmental effects clustering geographically, and they're hard to disentangle. So in this paper, we looked at whether these gene-environment correlations, whether they extend beyond the family, or were before this, many studies that showed that polygenic scores predict the family environment as well as genetic effects. And we looked at that here. So that's passive gene-environment correlations. You get born into a family, the parents pass down their genetics, but also the rearing environment. But what we also saw was that as these siblings grow up, the sibling with the higher polygenic score is more likely to move to a region with healthier environmental influences. So that's active gene-environment correlation. And what we also did, we ran GWASs on more than 50 traits related to physical and mental health and a lot of other stuff, and controlled for where people were born or where they migrated to, or both. And we saw that the genetic correlation with educational attainment decreased after controlling for the geographic region for almost all of these traits, most strongly for BMI and substance use, so traits related to what you put in your body. And this shows a similar effect, this figure. So if you focus on height here, you see that on an individual level, polygenic score for educational attainment explains about 1% of the individual differences in height. And the polygenic score for height explains 21% of the individual differences in height. It's pretty good polygenic score, but not the one that Loic talked about. But when you take the regional average of height and of the polygenic scores, then suddenly educational attainment outperforms the polygenic score for height, because on a regional level, this polygenic score also predicts these regional environmental differences. So here's the map, the actual map of the height polygenic score, and then the map of the phenotypic height. And those are not the same. And here is that polygenic score for educational attainment again. And you see that if it was all genetics, then the people from those coal mining regions should be the tallest of the country. But now they seem to be among the shortest. So these environmental influences that cluster with these genetic effects that are associated with educational attainment, they influence a very wide range of traits, and also educational attainment itself. So if you control for the region of birth or of current address for educational attainment, you see the SNP-based heritability substantially decrease. And that is, I think, because we, as a society, we make these genetic effects stronger. We reward certain genetic propensities of the traits that we value in society with a better environment, and leave the people that lack those propensities or have a lower propensity with a worse environment. So if you see that a child does well in school, he gets sent to a better school with better teachers and is more likely to grow up and find a better job and be able to afford to live in a better neighborhood. And in that neighborhood, he's more likely to meet a spouse who has the same genetic and economic luck. Assortative mating also shows this, the strongest effects for these signals. And over time, this could make society more unequal and also make our jobs harder, makes studying genetic effects more difficult, because these genetic effects and environmental influences are very difficult to tease apart. Just controlling for the region that people live in is just a very rude proxy of someone's social environment. So we have to think carefully about how we apply these signals, these polygenic signals. So this is one of the more controversial applications of polygenic scores, screening embryos in IVF treatments for their polygenic risk. So this is the first baby that has been born from such a procedure. Her name is Aurea. It says here, Aurea doesn't know it yet, but her birth was very special. She is the world's first PGTP baby, meaning that she is statistically less likely than the rest of us to develop a genetic disease or disorder throughout her life. So this is from the frequently asked questions section from the website of the company that assisted in her birth. It says, does genomic prediction screen purely cosmetic traits? No, we only provide risk scores for polygenic traits related to disease. And the other question is, does genomic prediction clinical laboratory screen embryos for increased intelligence? And the answer is no. So I took GWAS for all of the traits that they screen their embryo for. So you have brain disorders, heart diseases, cancers, and inflammatory bowel disease, and type 1 and type 2 diabetes. And I computed the genetic correlation with a whole range of non-disease traits, so anthropomorphic traits like height and BMI, reproductive traits, substance use, some psychological dimensions, and educational attainment and IQ. And of course, it's no surprise that these genetic correlations are significant all over the place because there is a lot of pleiotropy that was discussed yesterday in length. But we're starting to see that this pleiotropy does not happen on a just strictly biological level, but also through our environment, through the social environment specifically, through the way we organize our society and the way we create social inequality that explains a substantial part of these genetic correlations. So I want to thank my wonderful colleagues. I'm very happy to work with them. Some of them are sitting in this room, so thank you very much, and thank you for your attention.
Thank you. You briefly mentioned assortative mating. I was just wondering, to what degree is this geographic clustering just tracking the degree of assortative mating across those different traits?
Yeah, I think I'm not sure to what degree exactly, but I am pretty sure that those two are related, because if they cluster, they're also more likely to meet each other. So yeah, we're planning to look at how much of assortative mating is influenced by this geographic proximity. Yeah, that's something we have to look at empirically, but it's a very good question. Those two are closely linked, I think. Yeah.
I have a quick question. I was wondering, given the educational systems in the UK and Estonia, how do you think that these results would translate to like the US, where the educational system is sort of very different in terms of, educational attainment and who has access to higher education?
Yeah, that's a good question. I think, I suspect that it's going to look similar. A lot of the migration here is also related to education, either education or other economic reasons. But that's something we'll have to look at empirically. I would love it if someone that has access to these big data sets in the US could have a look at it. Peter.
Yes, so a lot of your results show there's gene, as you call it, gene-environment correlation or covariance, and that if you have, sort of population studies like GWAS, that influences your estimates of your and the interpretation of your associations. But what you didn't mention is that there are experimental designs that break that up. If you look within families, for example, and people obviously have been doing this and will do more of it, and you can see for those traits enormous differences in effect sizes, you know, within family versus population level. So that's quite interesting, I find.
Yeah, I think so too. Yeah, that's true. And they also they see a similar decrease in the heritability of traits that are closely related to educational attainment, but they also see for other traits that their genetic correlation with educational attainment drops when you look at within family, similar to when we controlled for region, which is effectively a within region GWAS. Yeah. Her?
Lindsay Fernandez-Rhodes, Penn State University. Thank you, Abdel. I really appreciated that you, for the first time, brought up life course perspective by looking at birthplace and then current address. When you plotted it out, you could look at the various categories and how certain groups would be differentially affected. Have you done something similar looking at socioeconomic status or educational attainment of a parent compared to the individual once they reach adulthood, and compared those, further kind of deepening your life course understanding of what's going on?
No, I haven't done that. It's a bit more challenging to do that in UK Biobank because there is not a lot of parent-child observations. But Michel Nifari has looked at in MOBA, who have collected parents and their children. And yeah, he's going to sit here later. Maybe you want to comment on it. I don't know what you found, or.
Like, I think you're right. We should do a lot more of that. And when you bring in like parents, but also parents of parents, there's like a whole new set of association tests you can do, right? You can like think about what Peter said. You can do it within sibling association, and you could also do within sibling association stratified by region. So there's like combinations and crosses of those kind of things you can do for like regional differences with. Rosa Cheeseman did some really cool work on that in MOBA, where they have parents and offspring, like 100,000 trios, basically, where you do both controlling per gene environment correlation using the parents, but then you can also do the interaction with regional things. And, you know, as we all know, you know, estimating either correlation or interaction just risks the other one, like mucking everything up.
Sorry, it's my fault to run late.
Yeah, we have 10-minute break, so back at 10:57. Yeah.